Bednarz-Knoll from the Medical University of Gdańsk. Today I would like to present the results of our research aiming to identify metastasis-initiating cells in prostate cancer patients. As you know, circulating tumor cells, CTCs, are seeds of metastasis. Some tumor cells within primary tumor might acquire aggressive phenotype and actively or passively enter bloodstream or lymphatic vessels. Such cells, if survive shear stress and immune system attack, might extravasate and in the new organ stay dormant even for several years, or immediately grow into overt metastasis. There is still little known which disseminating tumor cells are carrying highest metastasis potential. Therefore, the aim of the study was to identify putative metastasis-initiating cells in prostate cancer among other circulating tumor cells. To reach it, we collected peripheral blood from 118 non-metastatic prostate cancer patients, isolated mononuclear cells fractions putatively containing CTCs using density gradient centrifugation, stained them immunofluorescently for a panel of different inclusion and exclusion markers, and next analyzed using imaging flow cytometry, novel and powerful method newly implemented in CTC field by our team. First, we used immunofluorescence staining aiming to detect epithelial and mesenchymal cell markers, ergo common epithelial CTCs, and putatively also those in the broad spectrum of epithelial mesenchymal transition, known to indicate higher aggressiveness in many types of solid tumors. In total, we identified almost 600 potential CTCs of different phenotypes in almost 20% of investigated prostate cancer patients. Frequently, in one patient we detected coexisting phenotypes of CTCs, but only those of putative mesenchymal phenotype occurred to be associated with a shorter time to biochemical recurrence in multivariate analysis. As none of those CTC phenotypes correlated to metastasis, and mesenchymal markers including vimentin used in our study are usually not specific enough to detect tumor cells, we profiled matched primary tumors from patients with different CTC status using nanostring technology to seek for better markers to identify clinically relevant CTCs. Those patients whose tumors seeded putative CTCs of mesenchymal phenotype were among others more frequently characterized by higher EGFR expression. Therefore, we performed second immunofluorescence staining in the same patients, including detection of EGFR combined with androgen receptor, both potentially druggable proteins. Using this protocol, we detected CTCs in almost 30% of patients, but EGFR expression occurred only in part of them. And interestingly, only extremely rare CTCs characterized by high EGFR expression and the absence of androgen receptor were associated with the shorter time to metastasis occurring after radical prostatectomy. This interesting observation was consistent with our previously published study, where we showed that EGFR overexpression is found in a part of prostate cancer patients, might be associated with the higher stiffness of tumors, and is enriched in bone metastasis. To briefly summarize the study, we proved that similarly to primary tumors, which are highly heterogeneous, prostate cancer dissemination varies significantly both in yields, phenotypes, and clinical relevance of detected CTCs. Putative mesenchymal CTCs were indicators of shorter time to biochemical recurrence, whereas those characterized by EGFR expression and absence of androgen receptor were associated with occurrence of metastasis. I would like to sincerely thank all patients who agreed to participate in the study, as well as my team and all clinicians involved in this project. For more CTC images and also potential collaboration, I warmly invite you to visit our newly developed CTC Atlas, both present at webpage and LinkedIn. Thank you for your attention.